Great. Welcome, everyone. I know some folks will still be joining us as it is the lunch hour and it's just noon, but we'll go ahead and get started so we leave ourselves lots of time. As you just heard, the session is being recorded, and we will make sure that we post this session on the CitSci Alberta website for future viewing, along with the previous webinars that we've hosted. So we have been hosting this great webinar series on connecting people and science. Today's theme is on place-to-face partnerships, and I'm very excited that we have Dr. Hanington joining us from the University of Alberta to talk about his work. My name is Bradley Peter. I'm the Executive Director of the Alberta Lake Management Society. I'm also a member of the Citizen Science Alberta Community of Practice Steering Committee. I'm currently the chair of the committee, and I'll introduce you to some of our other members in a second here. I'll be the host for today's webinar, and then I'll be passing it over to Catherine to guide us through the Q&A for today. Of course, it is important that we take a moment to do land acknowledgement. And as a provincial organization, our members are from across the land that we know as Alberta, which is, of course, the ancestral homelands of the First Nations, Métis, and Inuit people. So we have Treaty 4, Treaty 6, Treaty 7, Treaty 8, Treaty 10, and 22 Métis districts across Alberta. Alberta is where Indigenous people have met, gathered, hunted, and stewarded this land long before the arrival of settlers. We recognize that this relationship between Indigenous communities and the land continues to this day. Traditional knowledge and ways of knowing of the Indigenous people are an important component of citizen science, and we are, of course, committed to finding opportunities to combine Western science and traditional knowledge in our practices. For those who don't know much about the Alberta Citizen Science Community of Practice, we are a collection of like-minded individuals guided by a very much volunteer steering committee. And our goal is really to grow the community of citizen science practice in Alberta, build out capacity and tools for delivering and participating in citizen science, and make sure we're enabling the use of citizen science data. So I encourage you to check out our website and take a look at some of the previous events that we posted. We've had lots of really, really great webinars. Here's a quick look at our steering committee. Again, I'm the current chair of the committee, but we are comprised of some really great folks from really great organizations: Battle River Watershed Alliance, Hanington and Area Land Trust, EPA, Misaki, the Office of the Chief Scientist, Nature Alberta. Got lots of great representation there. And if you think this is something that would interest you, please reach out to us and let us know. We could always use more help on our steering committee. So if you do want to reach out to us, here is how you can contact us: info@citscialberta.com or on social media, or of course through our website. So we do have a couple of poll questions we'll be sharing on your screen. Catherine will start the poll, and it really helps us to get to know who's in the room today. So if you can take a second to respond with what is your affiliation and what is your primary area of focus, that would be great. If you need other options or just want to type it in the chat if the poll's not working for you, you can definitely use the chat to do that. I'll give you a second to fill that out. Okay, Catherine, if you want to go ahead and share the results of the poll. Great. Okay, interesting mix today. Typically, I would say our biggest pool is the nonprofit pool, but today we have a lot of government representatives, which is great. A lot of water and biodiversity folks in the room as well. A lot of folks had others, so I'd be very interested to know what that encompasses. So if you chose other, maybe throw in the chat there what category you best fit into. That would be very interesting to see. Glad to see the water folks are represented today, especially given the topic of today's webinar. So glad to have everyone here. I see we've got some private consultants, folks who work on fish health, on stewardship. That's awesome. Diverse group today. Great. The webinar series was really designed to lead up to this big in-person gathering that we are hosting next Wednesday, October 30th. So if you're not registered for that, I encourage you to register. There's still time to do that. In 2018 was when we actually had our first gathering, which resulted in the Citizen Science Principles of Good Practice document. And so this upcoming in-person gathering is really building off of that work to celebrate the achievements of our community of practice, to create networking and learning opportunities, strategize around the use of citizen science data, especially as it relates to informing policy. And then I'm also really excited that we have two speakers joining us. So we have Krista Tremblett, who is a senior manager with the Community-Based Monitoring and Knowledge Unit, Airshed and Watershed Stewardship Branch with Environment and Protected Areas. And then Dana Duke, who is the Executive Director of the Mustaki Institute. So they'll be leading us through a conversation around how citizen science is driving change. And I'm also excited that we'll be presenting our new digital collaboration hub and getting some feedback from attendees on that collaboration hub. So there's lots to cover in that one day. It's a free event. So again, feel free to check that out on our website and register. So just a quick note about our agenda today. Our speaker, Dr. Hanington, is going to present for approximately 40 minutes. And then our steering committee has a couple questions based on the theme of this series. And then we'll open it up to Q&A from the participants. So you can put your questions in the chat box and I will be collecting those for Catherine. And then Catherine will be facilitating the questions to Patrick, and we'll try and get to as many questions as possible. So it's my pleasure to introduce today's speaker. Dr. Hanington is a professor in the School of Public Health at the University of Alberta, and his research aims to advance our understanding of organisms and our aquatic ecosystems by connecting DNA-based surveillance with communities and schools and industry and governments. And through this participatory research approach, Patrick and his research group have cast an Alberta-wide net to advance our understanding of microbes that impact recreational water or invasive species or species at risk or pathogens, a whole suite of organisms. So we're really excited to explore the importance of place-based partnerships with Dr. Hanington, and we hope you will find his that the insights shared are relevant and inspiring, and maybe you'll decide you want to be a partner in this project too. I'm sure Patrick's overwhelmed with partners right now, but that's probably a good problem to have. So without further ado, I'm going to stop sharing my screen and I'll turn things over to Dr. Hanington.
Great. Thank you very much for the introduction, Bradley. And hi, everyone. I also recognize quite a few names on the list of participants, so it's great to see people I know and people I don't know as well. Can you see everything okay?
That looks really
awesome.
Good.
Okay, great. So thanks everyone for joining today. Like Bradley just said, my hope today is I'm going to talk to you about some of the work that we've done on trying to bring molecular biology out to community partnerships and in a sort of a community-based monitoring context. So I'm going to talk about the work that we've done to, we call it democratize DNA. And I'm going to focus on two different stories to sort of emphasize some of the work that we've been doing. One is sort of a story that's just wrapping up, and another one is a story that we're about just getting started, really. And before I do that, though, I thought I would talk a little bit in background on a few things that I think are relevant. So it's probably don't need to tell this audience that there's a lot of different ways that community partnerships and water monitoring have been married together in the past. And a lot of the sort of traditional community-based monitoring or CBM gear shown here on this slide are tools that have been used in historical contexts and currently in partnerships with citizen scientists and community partners with academic institutions or government organizations or nonprofit organizations. These are often tools that provide us information about some of the basic characteristics of our aquatic ecosystems, like depth and turbidity, presence or absence of different sort of algae or blue-green algae, a variety of different chemical and temperature parameters, zooplankton. We can look for things like invasive mussels this way using substrates, observational data, and now more and more online data, which we've participated in as well with our work on swimmer's itch. But there are lots of great examples like eTick and iNaturalist come to mind as tools that are now more and more being used by community partnered research projects. And it is to the point now where even really sort of formal organizations like the US EPA even have handbooks on how to sort of craft and create citizen science projects that often see partnerships between governmental organizations like the US EPA, academic institutions, and community groups with varying interests in aquatic ecosystems. And so this handbook has evolved over time, but it is essentially a mechanism to try to provide some foundation for quality assurance and documentation of work that's done by community partner organizations. And the first topic that I'm going to talk to you about today is really on this idea of quality assurance, but I'll introduce you to that in a minute. As I have got more and more integrated into working in community partnered research, the sort of different terms that have come up frequently, things like citizen science, community partnership, participatory research, have all sort of bubbled up. And one of the first questions I had was sort of, when is our first use of some of these terms? And so when you look back in history, you can see citizen science emerges as a term sort of in the late 80s. It is defined by the Oxford Dictionary as scientific work undertaken by members of the general public, often in collaboration with or under the direction of professional scientists and scientific institutions. And I'm not sure I 100% agree with their definition, but it is in the dictionary, so I won't spend my whole talk criticizing it. It actually first appears in print, though, in 1989 in an MIT technology review. And I just think it's very appropriate that this first mention is in the context of an Audubon Society acid rain study, which has roots in environmental investigation on the impacts of acid rain on migratory birds. And so while this is certainly probably not the first time that citizen science or community participatory research was actually undertaken, I think it's interesting that the term itself is relatively recent, although maybe depending on when you were born, that doesn't seem recent, but it's within my lifetime, and so I feel like it's recent enough. If we look in the literature, though, the academic literature, over the last 30 years, there's been a really substantial increase in the number of scientific articles that have been published that mention some of the terms I've shared with you. So community-based monitoring, citizen science, and participatory research. The scales here are just over the last 30 years, and this is just a relative axis on the y-axis showing you the number of publications in the scientific literature that use these three different terms. And so you can see that really in the last 10 years, the incorporation of community partnerships in academic research has really exploded. And I think this is for a number of different reasons. If you read these papers, a lot of them highlight these sort of three really important dimensions that community partnered research adds to traditional scientific investigations. One is that we tend to see a real expansion of the capabilities of a traditional research lab, both in terms of the sampling capacity for environmental studies, the number of samples increases, the number of locations that can be sampled increases, and the number or the frequency at which those locations can be sampled increases. And because of those increases in sampling capacity, there's often a much more substantial incorporation of experimental controls, which is great for scientific investigations, which often really, especially at the environmental stage, often struggle to find appropriate controls for experimental type questions. So for example, we can sample more natural occurring water bodies and compare those to reclaimed wetlands, for example. And the more naturally occurring wetlands that we have in our study, the better our control is for understanding what those natural wetlands might look like for whatever parameter we're investigating. We also see study design can be impacted by community partnership. And those studies that often have successful partnerships typically address issues that are of both academic and public interest. And so there tends to be larger uptake for these types of studies, what we might call more applied research questions. And there's often a lot more interest in the results, both from certainly from a community perspective, but also potentially from a regulatory or government perspective as well, because there's that public interest dimension. And then finally, one of the really interesting sort of advantages of community partnered research is often that there's an advancement in sort of the scientific and water literacy and education within the public that participates. And so there's this much closer interface and communication stream that occurs between the academic organizations, the government organizations, and the partners that are in the communities. And that allows for there to be a much more free sharing of ideas and questions, and increases our ability to understand what's going on. Now, that all being said, there are sort of certain types of environmental research that tends to be very amenable to community partnered projects. I've shown you a few of these types of tools on my previous slide, but things like CitSci depth assessments and hydrolab assessments of temperature and chemistry, grabbing water samples and maybe doing some zooplankton tows, things that are really associated with techniques like water sampling, depth and basic measurements of the water, observations of animals and plants that are in our water bodies, and in some cases, online engagement and things like iNaturalist. These are areas that we tend to see very frequently in community-based research and monitoring efforts. However, there is a barrier that tends to be there when we start thinking about trying to move community-partnered research into some of the areas that are sort of the more quickly evolving, more advanced techniques that are used in an academic or government context. And so these techniques that tend to be more difficult for community-partnered projects to access typically include things that fall under sort of many of our regulatory guidelines. So for example, this is an image of the cover of the Alberta Safe Beach Protocol, which probably, I mean, all of you have read that, I'm sure. In that Safe Beach Protocol, we have protocols surrounding how beaches are assessed for water quality, things like assessing for fecal pollution as an example. But those techniques that we use to test for fecal pollution would have involved historically culturing bacteria potentially in a facility that allows you to culture bacteria, or now using techniques that are DNA based to assess whether or not certain fecal bacteria are present or absent in the water. So those techniques and even interpreting some of these protocols is sort of a barrier for community partnered research, because the techniques themselves are inaccessible to people for their use, and often the ability to sort of start formulating projects around things that are in these complex protocols is difficult to initiate. There's also a barrier to accessing some of the really cutting edge and cool research that we see in some of these more recently published scientific articles and that you might see in some of the more public-facing magazines around environment and ecology research. So environmental DNA, which is a technique that's emerging very quickly and evolving very rapidly around assessing for things like invasive species or species at risk, or assessing biodiversity, particularly in aquatic ecosystems, is a technique that's very exciting, has a lot of potential, but is something that is very difficult to sort of crack into if you're a community partnered researcher or a community group that's interested in trying to have this kind of work done. And it's actually in this area and essentially breaking down this barrier, which is what I want to talk to you about today. That's the focus of the two stories that I want to tell you, is about how we've been trying to move these types of techniques that typically are thought of as being sort of inaccessible to community partner projects over into this accessible category. To do this, we've worked over the last decade now, which, I mean, it seems shocking to me that the image that Bradley showed you was actually taken about a decade ago, and I still use it because I like it. And it also doesn't have any of the gray hairs that I've now gotten on the sides of my hair. But for about 10 years now, we've been working with community organizations, including ALMS and other groups in Alberta, on a bunch of different community partner projects. And a lot of our work is focused on a technique, quantitative polymerase chain reaction, which I will introduce you to in the next slide, into water monitoring projects. And so this is has taken a number of different forms and looks a lot of different ways to us, depending on our partners. So sometimes we see people collecting water samples in nice clear water lakes, diving in water, collecting things at the bottom. Sometimes we get to go out on boats. That's infrequent for us. Sometimes we're collecting water for fecal pollution at beaches where there's a lot of birds and poop. And we've worked on setting up garage-based laboratories for doing DNA-based analyses with people of a variety of different age groups, you can see here. And we've integrated some of these projects into classrooms for high school and junior high students. So we've tried our best to partner with organizations and individuals that have allowed us to work in a whole bunch of different really interesting and challenging, but also very sort of opportunity-focused contexts. And like I said, all of this work has really focused on trying to understand more about our aquatic ecosystems using this technique, qPCR, which, like I mentioned, stands for quantitative polymerase chain reaction. It's really a DNA-based approach that has emerged as a very, very now routine method that's being used for water monitoring projects. I'm not going to go into a whole bunch of detail on how this works. I just want to give you the high level sort of 30,000 foot view of what qPCR actually is. But it's based on this premise that organisms, like larger organisms like fish and other macro invertebrates and birds and things like that, all leave transient biological signatures in their environment that can be left in the form of feces or mucus or tissues when they die, for example. All of that deposits tissue and cells into our aquatic environments that then eventually release DNA into those aquatic environments. And at the same time, our aquatic ecosystems are often populated by several different microscopic organisms that also can be captured in a water sample and also have DNA. So pretty much anything that's alive, well, anything that's alive, not pretty much anything that's alive has DNA. And a lot of that DNA is unique to that specific organism. And so using techniques like qPCR, we can capture the DNA from an aquatic ecosystem. And using techniques like PCR, we can detect specific pieces of DNA that are unique to the individual organisms we want to know more about. And that allows us to create large sort of comprehensive projects that really focus on single water samples from an aquatic ecosystem. And from that single water sample, we can learn a lot about all of the different organisms that are present in the water. And so that's sort of the high level view of what qPCR is. I don't want to labor that process, but suffice it to say that over the history of this technique, which has been around now for over 30 years, it has often been a technique that requires these sort of larger, quite expensive pieces of equipment that you often only see at academic institutions or government laboratories. And even in those places, sometimes their costs have been prohibitive to being incorporated into more traditionally used monitoring efforts. So like I showed you that Alberta Safe Beach Monitoring Protocol, it wasn't until 2019 that qPCR was integrated into our recreational water quality monitoring efforts as a technique, even though it existed and the tests that we use in that protocol have existed for longer than that protocol has used them. However, as you know, with most technology over time, especially recently, things change very quickly. And these pieces of equipment that typically would have cost over $100,000 for somebody to acquire can now be scaled down, and the technology is much more accessible and affordable. And so these are examples that some of you, maybe even, especially those who've worked with us in the last 10 years, would recognize. These are smaller scale quantitative PCR machines that can do exactly what this big machine is doing on just a smaller number of samples in a slightly more simplistic way. And so as this technology has become more accessible, we've really been curious about whether or not we can use it in a community partnered context. And so that brings me to the two stories that I want to tell you about that both focus on different ways that we've tried to use this technique and some more advanced DNA-based techniques to understand more about our aquatic ecosystems and determine whether or not community partner research that uses these techniques can be relied upon. So the two stories that I'd like to talk to you about today focus on two different areas of research that we've been working on in the last 10 years. The first story focuses on developing a community and school-based qPCR water monitoring program. And I really want to just address this very foundational question here, which is, can we rely on partner-generated qPCR data? That's going to be the first story that I'm going to tell you about. The second one is going to introduce you to a newer project that we have going on right now, which is a much larger sort of multi-partner environmental DNA sampling project for invasive species and species at risk. And I'll get to that one in a minute. I want to focus on this first one highlighted in white first. And just to give you some context for what we're going to be talking about here. We're all probably familiar. Most of us are involved in recreational water, lakes in this province at some point. And so if you are, you've probably seen things like these at our lakes at some point. This is the two main types of water advisory that we tend to see pop up over the summer. One is a fecal advisory, and one is a blue-green algae or cyanobacteria advisory. These are two sort of health-related targets that we typically look for in our recreational waters, then that can prevent people from being able to enjoy those waters because their presence at a certain level makes that water become initially unsafe for use in a recreational context. And because of these two priority organisms, our first project, the one I'm talking to you about, really focused on trying to understand whether or not community partners could reliably test for these two different types of organisms in our water. Part of the reason that we focused on these was A, because they are often organisms that we get a lot of questions about, but B, it's also because they're organisms that we tend to find quite frequently in Alberta lakes. And so that makes them appealing targets for us to try to incorporate into a community partnered research question, because we don't end up with the problem that we sometimes find, for example, with invasive species, where we tend to find very infrequently or sometimes never an invasive species in any of our samples, which can be a little bit depressing for community partner researchers who are sometimes looking to just see if they're actually doing the technique properly. So this is a project that we were able to partner with a number of different organizations with. I'll introduce you to them at the end of the presentation. But we were able to bring qPCR to both community and school partners as part of this research project. And early on, as we were formulating this project, we came up with three primary models that we wanted to try to understand more about and how they worked within a community partner research context. The models essentially differ with respect to the level of engagement. And so, for example, in model A, which is a very frequently found type of model in the scientific literature, we have a situation where a researcher is designing a project. They're involved mostly in all of the data interpretation and all of the study sort of design components. And they typically are essentially utilizing community partners to help collect samples to expand their sampling size. A lot of the times, research projects that are focused on sort of app-based data entry from sort of unsolicited partners across a region fall within this model. A lot of the times, the data tends to reside with the researcher in this case. And if the community partners who are participating in the project want to learn more, they typically have to go and try to find that information after the researcher has analyzed the data. Model B kind of fits within the middle, where there's slightly more engagement and at least some level of data sharing. And then there's Model C, in which the researchers and the community partners co-design a research project. They are both involved in data interpretation, and there's a lot more integration down the road where the goal is really sort of a value share between all of the different partners. We realized very early on, and I'll reinforce this many times in the rest of this talk, that Model C is really the most sustainable approach to community partnered research. And certainly that's not any different for a qPCR-based project. Having that shared value and making sure that all of the different partners are getting what they need out of the research project or the monitoring effort is really important to provide that foundation for sustainability. So what we did is we very quickly decided that we needed to focus most of our efforts on trying to push Model C as often as we could. That often for us would mean that when we would find a potential community partner and would engage with them, it was often them that would choose, like, am I interested in cyanobacteria or enteric bacteria or both? And so often the choice of targets would be left up to the community partner, as would sort of some of the sampling plan as well, based on their local knowledge of the location. We ended up creating this very complicated sort of flowchart diagram, which is really not something I expect you to read here, but what I want to emphasize in it is that there were three categories of both development and programmatic implementation that we came up with. And so the color coding here is that the purple was the components of the project that was completed by the community partner on their own. This includes things like sampling, filtering the samples, extracting the DNA, running the qPCR reactions, and then entering the data into some sort of shared data sheet. The light, sort of the greenish color, was parts that were completed by the central laboratory. This involved sort of like developing the actual method for the analysis, so the assays themselves, and then essentially running parallel samples that were collected by the community partners to validate the data of the community partner. So our intention was to say the community partners is analyzing data, and we're going to analyze the same data and see if we come up with the same results as a way to validate their results. And then in the white is all the parts of the project that were completed in collaboration. And so what I want you to take away from this more complicated flowchart is that each partner had a very specific role within the study, and there were a number of parts where both partners were involved, including this sort of level of consultation and identification of monitoring goals, and then dissemination of the results, debriefing, and then essentially an evaluation of the project so that we can inform the next year. Ultimately, this project has led to two primary publications that are focused on sort of the theoretical component of incorporating qPCR into community partnered research. The first one, which we published a few years ago, you can see some familiar names on this publication, which is titled very similarly to my presentation today, Democratizing Water Monitoring. This was published by one of our first students studying this very earnestly, Sydney Rudko, who is now working within Alberta Health as an epidemiologist. And then the second, which is a paper we've actually just submitted for publication. Hopefully it will be sort of accepted soon. We're working on this one right now. This is from a talented master's student, Kaylee Welsh. And you can see people like Bradley are on these studies because they've ALMS has played a very integral role in some of this work. Generally, these studies look basically like this, where we have a water sample that's collected by a community partner. We usually get them to collect two different filters from that same water sample. And then each of those samples goes on to be processed, one of them by the community partner, and then one of them by us. And the one processed by us essentially uses the Government of Alberta beach monitoring test that's identified in that Alberta Beach Protocol. And then the community partner test runs a slightly modified version of that test on the qPCR machines that we set them up with. And so what I want to talk to you about today is a lot of work that's been sort of published or almost published in this study here about whether or not we can rely on community partner generated qPCR data. I feel like this part of this larger project really provides the essential foundation for us to move forward in a way that allows us to demonstrate that community partner generated data is reliable and can be trusted for use in other sort of secondary applications. So I want to tell you three things that we've learned from this sort of sub part of our larger project focused on identifying this sort of capacity of community partners to do qPCR. The first is that we developed a fairly comprehensive video and written training program that all of our partners would complete. And then we would do in-person training with those partners at least once to make sure that they were comfortable processing all the samples the first time around. And then we would essentially let them kind of become more comfortable on their own. If they ever had any questions, of course, we were there to answer questions. And different partners had different sort of levels of confidence in the application after we would sort of give them this initial training. But what I want to talk to you about today is a very specific part of the study where we just gave them this initial training. We gave them one round of sampling, and then we let them run samples. And so the three big sort of takeaways from this part of the study were that our community partner generated qPCR data met all of the qPCR quality criteria that would be in place for any official qPCR testing facility. As an example, the qPCR efficiency of our community partner data was 0.99, with one being a perfectly efficient qPCR run. And so that is really just to tell you the efficiency means nothing to you probably, but it's to tell you that they're basically as efficient as any core facility lab would be in running qPCR. So their technical capacity to run qPCR is just fine. The other takeaway message is that we found there to be a very strong correlation between our partner-generated data and the data that we would run using the official beach monitoring test. This is just showing you three of our inexperienced users, comparing them to Kaylee, our experienced user. And you can see here that each of these data sets essentially graphs the data points based on what did our inexperienced user find on the y-axis and what did Kaylee find on the x-axis. We analyzed this with a correlation coefficient, with a correlation coefficient of 1 being a perfect correlation. That would mean that the values were essentially exactly the same relationship between the partner data and Kaylee's data. Our overall correlation coefficient here in this whole study was 0.7, which is very strong. And it might be hard for you to see, but you can see that each of the different partners had slightly variable correlation coefficients. That's this Spearman R here, where we had around 0.84, 0.8, and 0.7 for some of the different partners. And so generally, we have a very strong correlation between the partner-generated data and the official tests that we would be running as part of the beach monitoring program, for example. And then finally, we wanted to look at data reproducibility. And this was essentially a metric that was one that we looked at because in the scientific literature, there's a study in which the US EPA sent the exact same water sample to a whole bunch of testing facilities across the United States. And what they found in that study is that there was a 0.66 log copies per filter variability between certified labs. That number doesn't need to mean anything to you other than that when we compared the same number between our community partner generated data and our expert generated data, what we found is that nearly the same value was present. So our partner data variability was between 0.67 and 0.85 log copies per filter, which is statistically indistinguishable from 0.66. And so the three big takeaway messages here are that the partner generated data is technically good. It has a strong correlation with any of the official data generated that as part of the sort of the protocol used in the beach monitoring program. And the partner generated data is just as reproducible as it would be as if the same sample was run by different core facilities that were ISO accredited. So this three big points here gives us a lot of confidence in the fact that that our partner generated data is very reliable. Then what we were able to do is apply this over the span of our entire project. And so in that project, we were looking at things like cyanobacteria, blue-green algae, enteric bacteria, and in some cases, some invasive species. And our overarching project engaged with over 120 community partners. And at the end of the day, we ran just under 9,000 qPCR reactions, and just over half of those were run by community partners. So out of all this data, then we generated a whole bunch of very reliable qPCR data about aquatic ecosystems in Alberta that we've been able to use to learn a lot about aquatic ecosystems at the same time as ways in which community partnered research using qPCR could be undertaken. So our takeaway message here from story one is really that community and school-based partners can very reliably participate in all of the steps that are associated in DNA-based water monitoring and research. We can trust them just as we can trust them to do temperature measurements or Secchi depth measurements. And in some ways, qPCR analysis is even more reliable than some of those other techniques because there is no interpretation that's done by the community partner. So the data that they generate can be analyzed by a research lab or a government lab, if that was the case. And that data then can be interpreted by an expert and doesn't rely on somebody determining whether or not they can see the Secchi disc at a certain depth or not. And so what's really important here is that what we take away from this is that the process of sampling, processing those samples, and then doing the actual qPCR analysis are all things that are accessible and doable by community partners. So that brings me to the second story that I want to tell you about. And this is a more recent project that we have focusing on multi-partner environmental DNA sampling for invasive species and species at risk. This is a project that's currently ongoing. And there's a few things that sort of served as our guiding principles as we initiated this project that we learned from the project I just told you about. The first is that all these relate to transparency and value, but this project, because it was multi-stakeholder, we wanted to make sure that everybody was really getting the value that they needed out of this project. So the first sort of guiding principle was that the goals and reasons that each partner is participating in the project need to be clear to all other partners. This gets at this idea of transparency. Everybody needs to know why everybody else is participating and what their objectives are in the project. And I think that was made very clear for all of the partners in this project that we needed to make sure that everybody knew what everybody else was hoping to get. All partners need to see value in continued participation if the project is to be sustainable. And that really gets to the idea I introduced you to earlier, this idea that we really need all of our partners to be able to identify their priorities and have their priorities be addressed in the project if they're going to continue to want to be part of the project. And so this again gets to this idea that we often try to let all of our partners choose their target organisms that they want to learn more about. And then we can build the research off of the samples once we've collected them. And then the third pillar here is bringing all partners and stakeholders to the table increases the likelihood of action. And this really gets to this sort of multi-stakeholder idea that this project was sort of formulated around, and it continues to grow actually. So it's gotten much bigger than what I'm going to show you here. But the idea here is that if we can bring to the table the community partners that are participating in sampling alongside government partners that are also participating in sampling and that are also trying to understand what that data means from a monitoring and a regulatory perspective, and maybe even designing policy around some of the results that are collected as part of the project, there's a continuum from sample to action, which really helps get everybody engaged and want to learn more and be part of the project. So this project that I'm going to tell you about includes a whole bunch of different partners. And like I said, it's changed a lot since its inception, but these are partners that are sort of now primarily part of the project. So you can see there's federal government partners like Fisheries and Oceans Canada, Natural Sciences and Engineering Research Council, Parks Canada. There are Indigenous partners. There are NGO, non-government associations and partners like Alberta Invasive Species Council, NSWA, Alberta Lake Management Society. There are other government partners like Alberta Innovates, the Alberta government, City of Calgary, and then there's our lone academic partner here, University of Alberta School of Public Health. And I think this is to really emphasize that all of our partners on this project make it possible. So we wouldn't be able to do what I'm going to show you here without having everybody be on board and essentially rowing in the same direction. So what we've done in this project is we've basically created a program that allows all these different partners to select invasive species and species at risk targets, and then go out and sample water that then is brought back to us for us to analyze for invasive species. This is what that looks like. So you can see here, some people get to sample in very nice aquatic ecosystems, like in the national parks. And then this is just to really show that sometimes I actually get out of my office. I don't always sit in front of the computer, and I did do some sampling. And this is essentially what that means is they're these little backpack samplers. They have a vacuum pump in them, and there's a filter, which you can barely see there, but the water gets sucked up through that filter, and that binds all the organisms and DNA onto it. And then this filter is what gets sent to us for analysis. And so you can imagine that what we've been able to do is essentially equip all of our partners with samplers like these that lets them go out and then sample locations that they want to know more about and tell us what they want to know about those samples. And our job as the academic partner has been to validate and develop new tests that allow us to answer their questions. And so what we've done is been able to create a panel of essentially, now it's over 40 target organisms. Some of them are included here. We haven't been able to draw them all into our little mini wetland anymore. So we've just added some more down to the bottom. But we have things like invasive mussels and invasive snails. We have Prussian carp and goldfish. We have various fish diseases like proliferative kidney disease and whirling disease, invasive invertebrates. We have some invasive plants like Eurasian water milfoil. And then we have a bunch of organisms that people care about protecting, things like lake sturgeon and Arctic grayling and Athabasca rainbow trout, cutthroat trout and tiger salamanders and long-toed salamanders. And then we have some health targets as well related to cyanobacteria and enteric bacteria like fecal pollution. So as part of this project, we've been able to really expand the number of target organisms that we can look for. And what we've done is, with all of our partners, going out and getting samples from various locations in different jurisdictions, we've been able to amass just over 1,500 samples over the last 2 years. You can see where all those samples come from on the maps here, but this is kind of a bit misleading because if you, for example, zoom in on Edmonton, you can see that there's this pretty extensive sampling that happens around through the Edmonton Storm Pond network and in and around the Edmonton area, and Calgary is very much the same. So these 1,500 samples all can then contribute to our understanding of invasive species and species at risk and health targets, whatever the partner wants us to look for. And just to give you an example of what that might look like, you can see here one of the main target organisms that we tend to look for a lot, especially in the storm networks of Edmonton and Calgary, and in between the Edmonton-Calgary corridor, are Prussian carp and goldfish. These are sort of the new hot invasive species in Alberta, Prussian carp especially being sort of isolated particularly to Alberta and Saskatchewan currently. And the Saskatchewan fish came from Alberta, so we're sort of the ground zero for Prussian carp invasion in North America. These fish are cyprinids. They look very much like goldfish that have lost their color. And once goldfish do lose their color, they become basically indistinguishable from Prussian carp. And so we've needed to work to develop DNA-based tests that allow us to distinguish these two species in eDNA, which we have done. We have a paper with many of our partners on it that we've submitted already for publication that talks about the development of this test. And as you can see in the next slide here, when we take the samples that were taken in 2023 and we look for Prussian carp and goldfish in the samples that were prioritized by our partners, you can see there's a whole bunch of samples that we've analyzed, and the ones that are in red are all ones where environmental DNA for either Prussian carp or goldfish were detected. And so there's extensive invasion of these fish within our storm ponds in Calgary and Edmonton, and even in the sort of central corridor between the two cities. And what's really exciting about this project is that because the eDNA gives us sort of an indication that something might be present, we can then go and try to ground truth that data by looking for the organisms themselves. And so this is a video taken by the city of Calgary that just shows you what that looks like here. If I can get the video to play, hold on. And so we can go to a storm pond and we can look, and you can see these little squiggly wigglies are all Prussian carp in this storm pond that's been drawn down. And so this helps us ground truth our data. It helps us add validity to the work that's being done. And as I showed you before, we know we can rely on these community partners to go and collect samples. And so this sort of allows us to close the loop on projects like this, where we can get a whole bunch of samples from a whole bunch of partners, analyze them for a whole bunch of targets, and then create research questions and add value to monitoring efforts for invasive species or species at risk throughout the whole province. So this whole project is really predicated on this idea that many hands make light work. And so the idea behind it is that we have this very cyclical approach to the research questions that we're answering and the monitoring effort itself. We have this partner-driven sampling and prioritization of targets. The processing of most of those samples is happening at the University of Alberta, just so that we have some way of archiving those samples and making sure that everything is stored properly. We then analyze the samples that were collected by our partners for the organisms that they targeted in their initial conversations. We communicate those results and we discuss the interpretation of those results with the partners that collected the samples first. And then once they've had a chance to see their results, we then share the results with the whole project team. This is sort of the phase 1 of the project, and then phase 2 kicks off, which is then the whole project team has a chance to provide continuity in terms of the sampling geography. And so then other partners can then identify secondary targets that might be of interest to look for in samples that were collected by other project partners. And so we can go back and retroactively look at those samples for secondary analysis targets, which then allows us to evaluate results of the entire program in a single sort of meeting, essentially. We can communicate project results to the team, and then that allows us to evaluate the results and plan for the next season. And so we've done this now. We've just finished our second season of collecting over the summer, and we have one more as part of this project. And now we're looking for sort of a sustainability to the program that allows us to hopefully continue to do something like this into the future. And so for the second story, the real takeaway here is that this idea of a multi-partner or multi-stakeholder participatory project really allows for specific partners to specialize, but also expand the team's capabilities beyond any individual member. And so we have this vision of multiple partners that begin to provide specificity based on their priorities and their own interests that focus us down onto specific individual questions for specific individual samples. But then those individual samples become a resource for all the other partner members in the project. And then that allows us to create a large scale, big picture data set that informs on invasive species and species at risk throughout the whole province. And as you could see on the map that I showed you, we're expanding into British Columbia and Saskatchewan and even Manitoba now. And just to give you a sense, all of that is being done by a single person here at the University of Alberta, Dr. Jacob Hambrook. So he's running all of the samples for all of the partners in this project. And so that gives you a sense of just how focal this little focal point in my simplified cartoon here actually is. It's a single person is running all those samples for all those targets. So I want to wrap up by just talking to you about some thoughts that we've had as we've done more and more DNA-based community partnered research. The first is that we've really bought in, we've drank the Kool-Aid on this multi-stakeholder partnership and its ability to build value for all the partners. We often think to ourselves, we're like a tiny little nesting doll in these larger questions. And to give you an example, on that map that I showed you of those 1,500 samples, we've actually only collected maybe 15 samples ourselves. And out of those 15 samples that we collected, it took us about two days, essentially, not including like all of the travel time to collect those samples. And so the network is really essential for us to be able to do this work. And as we sort of fit as this very small partner in this bigger project, the value continues to grow as more partners are added, and the capacity for that project to have value to everyone continues to grow as well. The other part is that participation really builds literacy. And we've seen this from guys who are retired participating in qPCR projects to students participating in qPCR projects. The more engagement and the more they can be part of the entire process, the more they think about how do we ask questions about aquatic ecosystems? How do we talk about aquatic ecosystems? And what should we be asking next about aquatic ecosystems? And that really gets me to the third thought here, which is that integrating the scientific method into community participatory research ultimately will always increase our confidence in the results that are generated by community partners. The study that I showed you where we compared our inexperienced users to our experienced user is a good example of that, where we really built that project around the idea of asking the question, can we trust community partner generated data? And to do that, we had to have a whole bunch of controls, and we had to have a whole bunch of considerations and blinded samples and all these kinds of things. And by thinking about it with that scientific method lens, we have created a data set that we're very confident in. And that allows us to then be confident in all of the other data that the community partners have generated, because we've been able to incorporate scientific method into our initial questions. And then finally, I think it can't be overstated how important it is that all of the partners are invested in the project. So the chain is only as strong as the weakest link. This is sort of the cliched analogy here. But it is very true that if you have partners that are in and out and in and out, and not always sort of completing the parts that they have committed to, it really makes it difficult to create something that's of value for everyone. And so making sure that there's that value add for all the partners is really, really essential. And with that, I want to wrap up by just introducing you to the three people who did all of the work that I just showed you. As I've introduced you to already, Dr. Sydney Rudko was sort of the student that I had who initially started this community partnership project and passed the torch to Kaylee Welsh, who's now just finishing her MSc. And then Dr. Jacob Hambrook, who is the one I mentioned, is the one who's the point person for all of our eDNA analyses and assay design for that project. So these three individuals are really the reason that I'm talking to you about this today. While I told you I get out every once in a while, it's not often I'm let out of the office. And then obviously there's been a whole bunch of partners from the funding and sort of organizational end that have been really critical for our work, as well as individuals, both sort of at the partnership level, people really critical, and then also all of the community volunteers, and this doesn't include all of them for sure, who were able to help us with all the sample collection and community partnered research that we're doing. And so I apologize for those not included on this list who did help, and just know that it would be multiple slides if I tried to get everybody on here. So with that, I will wrap up. I'm sorry I went a little bit long, so I'm happy to answer any questions for anybody, and I'll stop sharing my screen.
Patrick, thanks so much for your presentation today. That was great. It's really neat to see how you're working with all of those different partners and able to leverage those connections in order to really enhance your project and see value out of the work that you're doing. I have a number of questions in the chat and encourage folks to add more questions. I'm wondering if, though, I can ask one big question for you. This will be something we use in our in-person workshop that we're hosting on October 30th, and we'll be sharing these great touch points at that session in a video. And so what I'd like to ask you before I open up to all the other questions that we've had for you today, how is your work driving change?
Yeah, that's a tough question. I promise that I'll try not to ramble on this answer. I think, like I mentioned at the beginning, I think that community partner research is something that has existed within the environmental research space for a long time, and I don't think that those takeaway points that I have there are probably nothing that other community partner research projects haven't had to think about and address. I think that those are things that are probably very familiar to anyone doing citizen science and partnered research with communities. I think where we're maybe being more innovative is in thinking about community partnered research as if it doesn't need to be distinguished from the type of research that happens at a university, and trying to think of ways that we can apply those tools that we might consider sort of more at the cutting edge of environmental research to community applied research problems, and not being afraid to think of community partners as being just essentially inexperienced graduate students or postdoctoral fellows that might be at a university working on a project of their own. you know, I think that in that sense, it's probably the biggest change that we might be driving is a transition away from this idea that certain types of research can only be done in labs that have, you know, federal funded grants and have, you know, master's and PhD students running them. I think that what we've really tried to do is democratize that type of research to more people and find ways that we can train those people to generate data that is reliable to the point where then we can engage with them and provide some of that theoretical interpretation that might be needed for them to make sense of that data. So I feel like that's maybe the biggest change that we're hoping to push for here, is that we don't have to necessarily think of these things as being siloed as academic or government or in the purview of community science anymore.
Great answer. Thank you so much. I want to open it up. There were a lot of questions for you
Yeah, there are.
throughout the presentation, and
Gotta catch up.
and you covered so much so many different projects, and it was really good. So we'll just go in order in the way in which they came. But I also wanted to point out to folks that a couple of those great references that you referred to in your presentation, the articles that were written, I've included links that so that people can access those as well. Please don't hesitate to continue to add your questions into chat. Maureen Bell asked, DNA in organisms that are alive or have been alive, do hydrocarbons have DNA?
They do not. But she does, Maureen does bring up a good point that the DNA-based detection will detect both live and dead organisms. And sometimes dead organisms release a lot more DNA into the environment than the live organisms do. So those are things that are really important from the interpretation side, particularly for environmental DNA projects. Is, for example, a boat may have gone into a water body at some point where there were invasive mussels. And some of the DNA may be left on that boat, and then I might come in and I might take an eDNA sample right next to that boat, and it might not have invasive mussels on it anymore. Hopefully it stopped at the boat check stop and got decontaminated and stuff. But we might still detect invasive mussel DNA on it. And so that is one of the examples of the interpretation part that's really can be really helpful, is that, you know, we understand that that's the case, but if you're getting data from, say, you just paid for a company to run your mussel DNA sample analysis, you may not get that level of engagement that then they tell you, like, you know, you have to be a little bit cautious of what you might do with this data, because it might actually just be that it was a dead mussel or some eDNA that was left behind. But hydrocarbons do not have DNA, although they are pretty sticky and they tend to bind to DNA. And so if you have lots of hydrocarbons in, say, for example, a reclaimed wetland, that might be a good source of DNA, and it might preserve the DNA or keep it around so that you detect it a lot more long term. Yeah.
Thank you. From Sherry, on the school-based water monitoring, does this include water rangers?
Good question. It right now does not. A lot of our school-based sort of engagement happened through Inside Education and their outreach programs, with their programs like Navigate, which they did as sort of this biannual conference, and some of the educational outreach programs that they did. So that was really our exclusive way of engaging with teachers and students initially, and we managed through that to get in front of teachers during PD days and stuff like that. So I have 2 young kids, and I always wondered what happened to PD days, and now I knew that some of them at least were learning about qPCR-based water monitoring. Which was great. So yeah, that was where we were at with that. And it's actually the area that we're most excited about trying to expand more into is, you know, we really see the school-based programs as being sort of the sustainable long-term way that a lot of that infrastructure that we secured for that initial project can maybe continue to be used long-term in a school-based context. There's lots of places in the biology curriculum that are like really low-hanging fruit for us to incorporate DNA-based amplification of water targets while they're learning about DNA amplification in a cell and stuff like that. So, yeah.
Well, could I just add, so Water Rangers is a consistent program, so talking with them would be great. And then also the Battle River Watershed Alliance runs our extreme science program and would
Yep.
benefit from a partnership, so we should talk. But yeah, I am going down my own path. Frank had a question for you. Does the technology only work in an aquatic environment? For example, would an air or soil work just as well?
Yeah, so certainly in the environmental DNA space, water and like substrate sediment of aquatic ecosystems has been where a lot of the attention has been focused. There are air samplers that you can get now that essentially like just pull air through a filter, and then the air that will bind the DNA from the aquatic ecosystem or from the air. But there's also been a lot of comparator studies where they compare that approach to taking the water sample and looking for the terrestrial organism that may have left a signature in the aquatic ecosystem. So, for example, a deer might defecate near a water body, or a spotted lanternfly out in the Great Lakes area, which is a big invasive species they're worried about, will fly and just die because it lands in a pond somewhere. And what a lot of those studies have suggested is that the air is so dilute with the DNA that if you don't have really good spatial understanding of where your target organisms are, it can be a little bit difficult. But if you're using it as sort of a long-term surveillance effort, that it can certainly be used. So I think that that's sort of an area where there's a lot of advancement still happening. And the aquatic ecosystem, it's a much more robust technique at this point. Yeah.
Thanks. As Frank had a subsequent question. As costs drop, is there a risk that samples will be spiked to achieve an objective of the contributor? If so, how to mitigate this?
Yeah, absolutely. So, I mean, one of the things I didn't get into all the details of our community sort of qPCR program, but one of the things that we learned really early on that we just kind of had to contribute to the project was that, PCR is super, super sensitive. Like you can detect a single copy of your target gene DNA. So it's really easy to contaminate a space when you're running things like controls and things like that for your qPCR, because those are often very high copy number targets of your target gene. And so, in my lab, we have like absolute spatial segregation of like pre-amplification and post-amplification, and all this stuff is done in different spaces with like a whole bunch of tools dedicated to just decontaminating those spaces. When you set up a garage lab, you just don't have that. And so we would find immediate contamination of some of the satellite laboratories that we would set up in people's garages. So what we had to do was we would have to set up the controls ourselves in the lab here, and then give those to the partners for their use in their own analyses. So, I mean, Frank, to your question, like, yeah, absolutely. Like you might have nefarious business going on where people wanted to show that something was in their water, and then might spike something in to achieve the result that they want. But I would actually argue that it's probably much more likely that even they would just unknowingly contaminate the entire thing and then get everything positive and then not know how to kind of interpret the results, which would probably be likely and could also still have kind of a similar impact on the bigger project, where you might find that this whole location is positive because that one satellite laboratory or group, processed all the samples and contaminated the whole thing. And that was where, when we get onto the topic of reliability and trust of the community partner generated data, it becomes really, really important that kind of thing can't happen, because the second that you realize that 100 samples that came from, whatever lake were contaminated, then suddenly the whole thing starts crumbling, because then suddenly nobody can trust the rest of the data. There's questions about validity of the whole thing. So you really have to be careful with some of that stuff, because that can just erode the confidence in the whole thing. So yeah, good question. Yeah.
Yeah, thanks for that, Frank. Wendell Knowing asks, do you also carry out microbial source tracking with citizen partners? For example, if E. coli is found in a creek and it comes from humans, ducks, dogs, or cattle, I kind of know the answer to this because you and I have worked on this a little bit. But yeah, please, that's a great question. Thanks, Wendell.
Yeah, no, we definitely do, Wendell. Like, that actually is where the example I gave you, which was using the the target is Enterococcus, and it's the fecal indicator bacteria that we use in the province to indicate that there might be fecal pollution at a location. That test is actually not our preferred one to use in the community partner projects. We actually prefer to do the source tracking analyses because those are actually giving us more information about where the pollution is coming from, if it is there. And that's actually what we learned early on in the project, is that most of the partners weren't interested in whether or not there was fecal pollution. They were interested in where the fecal pollution was coming from. And so that was actually their question, not is our beach meeting or exceeding the recreational water quality guidelines. And often the places that we would be sampling weren't recreational locations. They would be, a spot outside of, like, under a bridge or something like that. So what we realized early on, and that was one of the big things that drove us towards this idea, that we had to test for the things that the partners really wanted to know about. We couldn't apply our desire for there to be an Enterococcus value on every sample because we didn't find any partners that really cared about what that value was in our community project. They wanted to know what was the source of pollution at different locations. So yeah, we did a lot of source tracking analyses for different partners. And then that comes alongside with a lot of requirement for us to assist in interpretation of that data, because that becomes a really sort of can be a bit of a cumbersome conversation to have when you're talking about, like, where's the human fecal pollution coming from here? So yeah.
Great question, thank you. From Frank, what is the barrier to participation, or the barrier to the participant? For example, if Joe Citizen is visiting a remote site, can they take a sample and send it in?
It's a really good question. Like, it kind of depends a bit on what the program is. So for example, a grab sample works just fine to do like a cyanobacteria or an enteric bacteria analysis. The key is in some of that is that it can't necessarily sit at room temperature in a bottle while you're, like, coming back from a backcountry hike, right? So there's a sort of a requirement for us to process the sample in a certain amount of time for us to be able to get reliable results. Then if we moved into the eDNA space, the same applies, but you also need to take a bigger water sample. So like a 200 ml grab sample is great for us to do a microbial analysis on, but we need like 2 liters to do an eDNA extraction on. And that's part of the reason that in that eDNA project, we've given the partners the samplers, the eDNA samplers. It makes the project more expensive to do it that way, but it also means that it's way easier for the partners to take 20 or 30 samples in a sampling run when they only have to get a filter out of that process, rather than like a 2-liter bottle of water that then has to get shipped to us to filter. We would have run out of money already just on shipping costs, shipping that much water around. So that's then the barrier for that kind of a project, is like, it is really helpful for the partner to have an eDNA sampler or a way to filter the sample, because it then takes the pressure off of having to get it to us really quickly. But generally, yeah, if there's people that are interested in these kinds of projects, there are ways to get them engaged and to have them be able to participate by collecting water samples. So yeah.
Thank you. Can you talk a little bit more about how the community partners use the information or the results collected from QPCR examples? Yeah, thanks.
Yeah, no, it's a good question. So one of the things that one really good example is we had a partnership with Moose Lake looking at cyanobacteria, and for especially over a period of years, there was a lot of questions about like what was the main sort of problem at Moose Lake related to cyanobacteria. There were like a couple of dogs had died. There was questions about like, where was the cyanobacteria toxin coming from? The dogs died during a time when like the analyses were suggesting that there were no toxin producing bacteria, and then it wasn't until a little bit later in the season that the lake was posted. And so in that case, like, it was very serendipitous, but we had an enteric bacteria project with Moose Lake at the same time, and then we got over 100 samples collected for enteric bacteria analysis at Moose Lake during the same year that the dog deaths had happened. And so we were able to then go back and retroactively analyze all of those samples for cyanobacteria, looking for the toxin gene. And we found specific locations along the lake that were very high for the toxin gene that encodes for the microcystin that comes out of the cyanobacteria. And so we were able to get a lot better spatial resolution because of this other project that had been going on related to fecal pollution. And so that's sort of like one example where like we're able to essentially provide an answer about fecal pollution to the lake. They get an idea of like these are areas where there might be hotspots, and we try to essentially, we try to drive the question to be something more around the idea of like creating sustainable data sets for our partners, rather than like pinpointing that Patrick's cottage at that lake is the reason that there's fecal pollution because he hasn't updated his septic system recently or something like that, right? And so we try to suggest that it's about the consistency and the spatial scale that we can accomplish with DNA-based testing and answer questions about numerous organisms that gives the real value, because we can provide these baseline data sets that then allow lakes to say, well, how has this changed over time? If something changes at the lake and a new development goes in or a farm moves in that is connected to the lake in some way, we can understand that, well, this is what the lake looked like for the last 3 years, and now it's changed to this new state with this change that's happened. So I think that's really the most value that we can provide, is that sort of like longevity and the archival of the data and the ability to go back in time essentially and analyze samples for targets we didn't know we needed to look at for certain things down the road. So yeah.
In a related question, can the data collected on invasive species on goldfish and carp be shared?
Yeah. I mean, a lot of that information is actually already public. So we've because we're partnered with Alberta Environment and Protected Areas and the Invasive Species Program. They actually have a whole page on goldfish and Prussian carp, which is actually like the ground truthed data, where they've actually found the carp. And that's like what we always would say is kind of like that's what's required of the eDNA program. Is we use the eDNA as a great way to help kind of like maybe prioritize those visual inspection efforts. We can say, hey, we got something here. It might be good to go and look. But we wouldn't use it as a way to say this location is positive for this organism conclusively. And so that example is really great, is that, then we have visual confirmation of these fish in certain locations, and that's all available on the Alberta Environment Protected Areas website. And then the eDNA project has been really great because it's allowed us to kind of confirm those locations and identify maybe new ones that need to be investigated. Yeah.
Thanks. Sasha had to run, but her question, and I think it's relevant to all of us, is how has the training content been developed, and how are you able to break down the essentials of research for those partnerships?
Yeah, the training content. So we actually hired a high school biology teacher to do a master's degree with us. And her name was Kelsey, and Kelsey actually essentially aligned all of the things that we needed to do in a molecular biology project to the high school biology curriculum, and then created a sort of a modular training program that included like a bunch of shorter videos of just like showing the method, and written training that kind of like emphasized the key points. And then we implemented that, and then we refined it like 6 times, because we found out that like all these different things would come up where, we wouldn't understand why something had gone wrong, and we would go and watch what they were doing, and we'd be like, oh, like that's what's going on. And so then we would say, okay, we got to update the training program to really emphasize this part of this now. And so that's where we got to at the point where we then gave all that to those partners that I showed the data for, is they had the sort of the finalized, refined version. And then that program actually went and was piloted in Michigan, where we do a lot of collaborative research. We found a college in Michigan, Northwestern Michigan College, that created a freshwater studies program, and we got them to use our training modules as part of a DNA-based sort of modular program that they incorporated into their program for 3 years. So we got to kind of like trial it out in an educational context with students that we knew we were kind of the guinea pigs. So yeah, and it's certainly not perfect at this point, but it is pretty good. I think what we found is that most people find it accessible, and that it's short enough that they don't lose attention, because the videos are too long and stuff like that. Those are all things we learned as we went, that like people don't want to listen to me talk, or any of us talk about DNA for more than like 6 minutes. So thanks for sticking around, everyone.
Yeah, there's still actually quite a few questions. We are going to wrap up the quote Q&A in just a few minutes, about 1:25-ish, but there are a couple more. These are just great questions, and it is really good to hear how you refine it and align it with the curriculum, because I think that adds value to teachers. It shows also just the flexibility of a project to sort of see how does it actually work on ground. Is it, are these students really interacting well with this, and are you getting the kind of results that you want? I like that. Frank asked, publishing in the science literature is important. How much should the results be democratized, in addition to the collection, for example? Conversely, what are the considerations to ensure that results are used appropriately and interpreted correctly?
Yeah. I mean, there's kind of, I think there's two pieces there. So I mean, from our academic standpoint, the publication side is sort of our currency, and it's how we kind of lend more credibility to the research that we're doing when we apply for grants and things like that. The challenge historically has actually always been that scientists generally are not trusting of community generated data, which was really the impetus for us to do the study that I presented to you guys here, to essentially demonstrate that we can rely on them to do this work. And so that's part of it. And I think that our philosophy, especially within our community partner projects, has always been that those publications have to be open access. So they have to be papers that people can access without having to pay anything to find them, which I could give you another hour-long rant about open access publishing and how it's a sort of a scam for taxpayers and stuff like that. But we don't have to worry about that right now. I think that it's really critical that it's open access. However, that being said, scientific publications are just by nature often not really accessible by community partners. The review process generally shifts a paper that might be accessible towards something that's much more concise and technical jargon, rather than being wordy and explanatory. And so we often get comments back on these papers that it's like, this is a word salad, there's too much here, you got to cut this out, make this more concise, blah, blah, blah. And by the time you get through all that, then the paper is basically just like any other scientific paper. And anybody who's not at least well-versed in the methodological jargon just can't access what we're talking about. That's where I think that the communication piece really comes in and is critical, is like, what we would see as being our role, maybe more important even than running the samples or kind of like coming up with some of these crazy ideas for projects, is actually on the communication end. How do we take the data that's generated by these projects and communicate it effectively to the partners? And honestly, that actually is probably where most of our time is spent. Like, we would get samples from partners, we would take them, run them, or teach them how to run them, and then they would get a spreadsheet with a bunch of numbers, and that means nothing to them. Like, they don't know what a CT value of 32 means compared to a CT value of 41, but that is very meaningful in terms of what the actual meaning of that data set is telling them. So it's the process of sitting down and communicating that and explaining it and getting it to a point where the data can be translated into something that makes sense, but also is not in a way like condescending to the fact that like all this stuff exists. So it's about kind of communicating that like, these are what these values mean. We don't have to talk too much about this, but this is how you would interpret it and what you would do. And then often they just want to know, like, what do I think about this? They're like, well, that's great, we found some human fecal pollution, what should we do about it? And that's like, it's often to that level of interpretation where they're like, well, now we've got it, what do you think we need to do? And so that piece is really, I think essential for there to be longevity and sustainability to the programs, is that you need to have that translation. And I think it's why a lot of these techniques are, they're not accessible if you get them done by a contracting company or something like that. If you want to get your, for example, if you want to get your well tested for fecal bacteria or E. coli at your cottage, you get a data set back and it's like, this is your colony counts per 100 mils of your sample, and here's your geometric mean. And everyone's just like, I don't know what this means. and there's not even sometimes like a way to interpret what that value is supposed to mean to you. So I think that's where the academic partner is really can be really helpful, but it's taken us a lot of time to realize that we have to communicate in a way that is at the level of the community partners and that makes sense to them, because we can't just come in and start talking about like, well, you know, you do this and you do this, and we can do this meta barcoding thing over here, and because that still doesn't get to the point of being accessible. So yeah, it's a really good question. So
Yeah, I just, because of the timing, we had one last question about water and the number of sites sampled in a single water body. When will results be available for this summer sampling?
Yeah, so like I said, we're at the stage where the first cycle of the 2024 data set has kind of almost wrapped up. So we almost have all the data from 2024 now. And then we share that with all the project partners first. So they get kind of like jurisdiction over their data. And then they can kind of like see what it says, we can interpret it with them. And then we move to sort of the phase 2 where we're going around the other end of that cycle. And then honestly, it becomes a situation where in some cases it's up to the partners about where that data and how that data is shared with the public. Our intention obviously then is at like different points within the program, our goal is to publish the data. So then we would share it in a publication format specifically, but it's a cumbersome thing to work with because we have all these different targets now that we're looking at. And so, you know, for example, we've never found an invasive mussel. So that's really not a exciting data set for anybody, except if you're the invasive species manager and you're just really happy that we don't have mussels yet. But for other things, it's like, well, what does this one little point up here mean? And so it's hard to get some of that information out sometimes. But yeah, I think part of our goal for the last year of the project is to try to figure out how can we make a public facing portal that allows people to kind of like screen the data once it's all been vetted by the partners and it's okay to share. So yeah, it's a good question, but yeah, it's coming. It's coming.
Oh, very exciting. Patrick, thanks so much for sharing your lunch with us and all of this great value of partnerships and citizen science. I think we learned a lot today, and I hope all of you have an opportunity to reach out to Patrick if you have a related project or possibility for a partnership. So thanks to everyone for attending today. And being here. We have an in-person session to wrap up our whole Connecting People in Science webinar and workshop series. Our in-person session is October 30th at the Bison Lodge in Edmonton from 9 to 5. We've still got a few spots left, so please join us. We've got some great presenters, as Bradley mentioned. Our sponsors for this event are the City of Edmonton, as well as the Office of the Chief Scientist. And we're hoping that all of you can come and help us expand on our opportunities for citizen science in Alberta. We can learn from each other. In addition, we're going to highlight our new hub. And that hub, Patrick, is an opportunity for groups, including researchers, to share their citizen science projects and create links to those that open science, open data sets, and also opportunities to make sure that information is available. So please come out, everyone, enjoy us and enjoy the great presenters that we have. We have a panel coming up with some lightning talks on their own projects, and we'll tap into all of your experiences in citizen science to discuss ways in which we can make citizen science data available to all the practitioners in the province. So hope to see everyone on October 30th. And Patrick, again, thanks for being here today.
Yeah, thanks everyone. Thanks for sticking around and for all the great questions.
That's it for us today. We'll wrap up and hope to see you on October 30th.
Bye, everyone.